The Disruptive Dozen is a rank-ordered list of 12 closed-to-market cutting-edge technologies that leading Mass General Brigham faculty feel will have the greatest impact on healthcare in the next 18 months. Number 10. Making cells larger to see them more clearly. Visualizing cells at high resolution is a cornerstone of modern biology and medicine. Over hundreds of years, as scientists yearned to observe biological structures with greater power and clarity, they built more advanced microscopes. Yet today, even the most sophisticated tools have limits. So, a team of scientists in Massachusetts took a fresh approach to this problem. Instead of improving the microscope, they developed methods to physically enlarge or expand cells and tissues of interest. This approach, known as expansion microscopy, is revolutionizing the way researchers examine, characterize, and diagnose biological specimens. For example, expansion microscopy has made it possible to visualize individual neurons and their connections within the brains of laboratory models, including fruit flies and mice. By marrying expansion microscopy with other state-of-the-art imaging methods, the Massachusetts-based team, together with their colleagues, achieved what previously could be done only with high-powered, expensive microscopes. They also visualized millions of neuronal connections in just a few days, enabling large-scale brain mapping studies that would otherwise take years to complete. Expansion microscopy is making inroads in the clinic, too. The method has been adapted for use with patient samples, allowing pathologists and clinical researchers to visualize fine, nanoscale structures in human cells, such as the fine projection of kidney cells, which are typically viewed using more complex, time-consuming methods. That means with expansion microscopy, pathology samples can be processed more quickly, which could help speed up diagnoses.
Even with all the recent advances in optical technology, we're still limited by the diffraction of light itself. I'm sure any pathologist like me can attest to the many times when we have wanted to go just that one magnification step higher to literally crawl into the tissue, but then find ourselves unable to do so. Expansion microscopy is a great tool for researchers and diagnostic pathologists alike that can help us to not only visualize, but also to annotate with molecular markers nanometric structures, such as, for example, the kidney filtration barrier or small organelles within cells. We can visualize disease processes that then enables us to make better and more precise diagnoses. Expansion microscopy has the potential to revolutionize diagnostic, pathology, and disease research.